Welcome to the Deep Dive. Our mission today is to cut through some of the noise in longevity science. We're zeroing in on three molecular mechanisms that are right at the forefront of fighting age-related disease.
And we're not just talking about symptoms here.
No, not at all. We're looking at the foundational processes, the things that actually cause cardiovascular decline and neurodegeneration.
That's really the crucial distinction, isn't it? I mean, aging isn't just a slowing down. It's this accumulation of molecular damage that leads to chronic inflammation, hormonal problems. To really extend your health span, you need to intervene right there.
Exactly, right at the source. And that's our plan for today. We have three main pillars for this deep dive, all pulls from the very latest findings. First up, the systemic effects of GLP-1 receptor agonists. These drugs are doing way more than we thought. Second, we're going to talk about the master regulator of chronic inflammation, a molecule called NLRP3.
The master switch, yeah.
And finally, a really surprising compound that your own body makes to protect the heart. So let's just jump right in.
Okay, so let's start with the hormones that have been dominating the headlines for the last couple of years. The GLP-1 receptor agonists, or GLP-1RAs.
Right.
I mean, the basic function is pretty well known by now. They're great for managing type 2 diabetes. They lead to effective weight loss and, improved cholesterol.
But the whole story has really shifted. It's moved from just metabolic management to genuine cardiovascular protection. We have multiple large trials and real-world data confirming this now.
And they reduce MEC, right? Yeah. The major adverse cardiovascular events.
They do. So that's your non-fatal strokes, heart attacks, and cardiovascular death. The benefit is especially clear in patients who are already dealing with T2DM or obesity.
But what's really key for you to understand is the mechanism behind this. For a while, everyone just thought, okay, they lose weight, their blood pressure gets better, and that's why their heart risk goes down.
But that's not the full picture.
Not even close from what I'm reading. Absolutely not. The data shows the protective mechanism is often totally independent of weight loss or glucose control. I mean, take alglutide.
An older one.
Yeah, an older GLP-1RA. It was only, let's say, modestly effective for weight or glucose, but it still reduced MPE by a huge 22%.
Wow.
Which just proves that these drugs are doing something directly. They're acting on endothelial function, reducing inflammation right inside the blood vessel walls. It's a benefit that goes way beyond just the metabolic improvement.
So that brings us to the big question everyone has. How do the new heavyweights, semaglutide and tirzepatide, actually stack up against each other in the real world?
Well, we can now look at these huge U.S. claims databases, which compare them in very large, diverse populations, and the data confirms, that both of them significantly reduce cardiovascular risk compared to older meds. It reinforces the power of the whole GLP-1 class.
But what about head-to-head? Semaglutide versus tirzepatide on that primary MPE outcome. What did we find?
They seem to be pretty comparable. The analysis gave a hazard ratio of 1.06 with a very tight confidence interval. So for that big composite risk of major heart events, they offer similar protection in day-to-day clinical practice.
So being a dual agonist doesn't automatically mean you get a double benefit for MC.
It doesn't seem to, no. But there was a really important nuance in the data, wasn't there? Something specific about heart failure.
That's right, the heart failure data.
Yeah, when semaglutide was compared to, say, a DPP-4 inhibitor like sitagliptin, it showed a much lower risk for hospitalization from heart failure. The hazard ratio was 0.61.
So a nearly 40% reduction in risk.
Exactly. And that suggests there might be a different, more direct benefit on the heart muscle itself, or maybe in how it regulates fluid volume, which is a major driver of heart failure. That's a huge area of research right now.
Okay, let's pivot from the heart to the brain, because this is where the whole anti-aging potential of these GLP-1RAs really starts to, expand.
It's profound, really. We now know GLP-1RAs have these strong anti-inflammatory and neuroprotective effects right inside the central nervous system.
How are they doing that?
Well, they actively suppress some of these chronic inflammatory pathways, especially NF-κB, which is a master switch for turning on inflammatory genes. They also shield the brain from oxidative stress.
So they're basically arming brain cells against the two biggest drivers of neurological decline. Inflammation and oxidative stress.
Yes, and they're doing it right where it counts. The GLP-1 receptor is all over the hippocampus.
The memory and learning center.
Precisely. And activating that receptor, which is what these drugs do, clearly improves learning and memory, We're seeing incredible promise in Alzheimer's models where dual and even triagonists seem to reduce cognitive deficits.
I think one of the best examples of this brain-first action is how they change eating behavior. It's not just a gut reaction, it's a rewiring of the brain. Tell us about those DMHGLP1R neurons.
Right. This gets to the core of the behavioral change. These drugs boost the activity of a specific neuron cluster in the dorsomedial hypothalamus, the DMHGLP1R neurons.
And these are tied to something called pre-ingestive satiation.
Exactly. It's the feeling of satisfaction you get before you've even really finished the meal.
So your brain is predicting it's full rather than waiting for your stomach to send the signal.
That's a perfect way to put it. The activity of these neurons surges when you're just accessing food, smelling it, getting ready to eat. After you take a GLP-1RA, that neural sensitivity is way up, so the brain decides it's had enough much, much faster.
Which means you eat less.
You eat less, and the whole process is being managed from the brain, not the stomach.
This all leads to maybe the most fundamental finding about aging itself, the body-wide effect. I'm thinking of that data from aging mice treated long-term with exenatide.
Yes, this is where systemic anti-aging really becomes the right term. These multi-omic studies found that long-term treatment actually counteracted the gene activity changes that come with aging.
And not just in one place.
No, across a huge range of organs. The heart, the kidneys, white blood cells, and again, the hippocampus.
Wait, you said the hippocampus specifically. Did it literally make the brain younger?
In a sense, yes. It reduced the epigenetic age of the hippocampus. It reversed some of the key molecular signatures of aging in the brain's memory hub.
That's incredible.
But here's the kicker, the most important piece of the puzzle. When the scientists blocked the GLP-1R gene just in the hypothalamus,
Just in that part of the brain.
that whole body-wide age counteracting effect was significantly reduced. It proves that the brain, that hypothalamic signaling, is the central command center mediating these anti-aging signals to the rest of the body.
So these aren't just weight loss drugs. They're a systemic anti-aging signal that starts in the brain.
That's what the evidence is pointing to.
Okay, let's move on to our second pillar. We're going to target the root cause of so many age-related diseases, what scientists now call inflammaging.
It's that chronic, sort of smoldering, low-grade inflammation that just fuels everything from heart disease to Alzheimer's. And the key target here is something called the NLRP3 inflammasome.
The master switch you mentioned earlier.
Exactly. Think of it as a sensor inside your immune cells. It's just waiting to detect danger signals, stress, dying cells, protein clumps, and when it does, it flips a switch that unleashes a torrent of inflammatory molecules.
So if you can turn that switch off, you could potentially halt inflammaging right in its tracks. And we have some exciting phase 2 clinical data on an oral NLRP3 inhibitor, VTX3232. How big was the impact?
It was dramatic. It really validates the whole theory. VTX3232 caused a nearly 78% reduction in HSCRP.
The classic marker for systemic inflammation.
After just 12 weeks. And crucially, it also brought down levels of interleukin-6 and lipoprotein, or LPA.
Okay, let's pause on LPA. For anyone listening, why is lowering LPA with an anti-inflammatory drug such a big deal?
Because LPA is notoriously stubborn. It's a cholesterol particle that's mostly determined by your genetics, and it's a major risk factor for heart disease that most of our standard drugs, even statins, can barely touch.
So the fact that an NLRP3 inhibitor can lower it is huge.
It's huge. It suggests that inflammation is playing a much bigger role in how LPA works or how toxic it is than we ever realized.
Now here's the insight that connects this back to the GLP-1RAs we were just talking about. This inhibitor did not cause any weight loss.
None at all. VTX3232 didn't cause weight loss on its own, and it didn't add to the weight loss from semaglutide. And that separation is absolutely vital.
Why?
Because it means we have two completely different complementary ways to treat this. The GLP-1RAs handle the metabolic side of things, the appetite, the hormones. The NLRP3 inhibitors go after the residual inflammatory risk that's still there, even when your weight and glucose are under control.
So this points to a new kind of therapeutic strategy.
Absolutely. We're looking at a future with a therapeutic triad. You'd have statins for lipids, GLP-1RAs for metabolism, and then NLRP3 inhibitors to aggressively shut down that chronic inflammation that other drugs miss.
And it seems like NLRP3 is involved in almost everything age-related. It really is a pipeline in a product.
It's incredibly versatile. In the brain, it's activated by amyloid plaques in Alzheimer's or alpha-synuclein in Parkinson's. In the heart, it's activated by cholesterol crystals. It's a player in kidney disease, fatty liver disease, you name it.
And we're already seeing clinical results in the neurospace.
Yes. Trials with inhibitors that can get into the brain, like NT0796 for Parkinson's, have shown they can lower inflammatory markers right in the cerebrospinal fluid. And more importantly, they lowered neurofilament light chain, NFL, which is a direct marker of neuron damage.
So we might finally have tools that can actually slow the progression of these diseases.
By stopping the inflammatory fire, yes. That's the hope.
For our final pillar, we're going to shift gears completely. We're moving away from new drugs and looking at an endogenous molecule, something your body makes itself, 12,013-dihome.
Right. This is a lipokine, which is a fat signaling molecule, and it comes from a very specific place, brown adipose tissue, or B-digame.
And the problem is, our levels of this molecule drop as we get older.
They do, in both humans and mice. And this decline happens right as age-related heart problems start to ramp up. The research shows that if you reverse this decline, if you boost 12,013-dihome levels, you can preserve heart function even in very old mice.
So it's like an internal repair mechanism that just runs out of juice. How does this tiny molecule physically rescue an aging heart cell?
It protects the cell's internal machinery from stress. As the heart ages, its cells build up something called endoplasmic reticulum stress, or ER stress.
The ER is the cell's protein folding factor, right?
Exactly. And when it gets stressed, it sends out signals that lead to cell death and scarring, or fibrosis. 12,013-dihome works by calming down that ER stress.
Okay, that makes sense. Less stress, less damage. But there was a second mechanism too, wasn't there? Something to do with muscle contraction.
Yes, and that's all about calcium. 12,013-dihome also dials down the activity of an enzyme called CaMKII. Now, CaMKII is critical for regulating the flow of calcium in and out of the muscle cell, which is what makes the heart contract and relax.
And it gets out of whack with age.
It can become hyperactive, which leads to inefficient or irregular heartbeats. By reining in that hyperactivity, 12,013-dihome helps maintain that perfect calcium cycling. It keeps the engine running smoothly.
So a very clean, two-pronged attack. Now, the biggest curveball in this research was about sex differences. This benefit wasn't universal, was it?
No, and this is probably the most crucial takeaway here for the future of medicine. The protective effects, the reduction in ER stress and fibrosis, were seen almost exclusively in the male-aged mice.
Why would that be?
Well, when the researchers looked closer, they found that the female-aged mice didn't really have a big increase in ER stress genes to begin with.
So they didn't have the problem that the molecule is designed to fix.
It seems that way. They may have some other, endogenous protection against ER stress that males don't have. So while the treatment helped the males a lot, it didn't do much for the females because they hadn't developed that specific pathology yet.
That's a massive finding. It tells you that the aging process itself is different at a molecular level between sexes.
Exactly. It completely moves us away from one-size-fits-all treatments. It tells us that for a male, you might need a 12,013-dihome-like molecule. For a female, you might need to target a completely different pathway to get the same benefit.
So when you put it all together, what does this all mean for the future of extending our health span? We've covered three incredibly powerful but very different approaches.
We've got GLP-1 arrays, using hormones to signal rejuvenation from the brain outward. We have NLRP3 inhibitors, turning off that master switch of chronic inflammation. And then we have 12,013-dihome, a targeted internal rescue for the heart.
It feels like we're getting so much more specific.
We are. The GLP-1 data shows us that targeting the brain can have these body-wide anti-aging effects. The NLRP3 data shows that if you hit inflammation hard enough, you can solve problems like LPA that nothing else could touch.
But like you said, to really use these tools well, we need precision. We know aging isn't uniform. Inflammatory genes might spike in microglia in the brain, but not in other brain cells.
That is the frontier, the single-cell level. Which brings us to a final provocative thought for you to chew on. What if the next generation of diagnostics could measure your biological age on a cell-by-cell basis?
So it could tell you that your hippocampal neurons are aging faster than your cardiac cells.
Precisely. It could identify which specific cell types are falling behind. If we could do that, how dramatically would it change how medicine uses these powerful targeted molecules? The future of anti-aging won't be about treating the average age of a person, but about fixing the specific cells that need it most.